Okay, so let's go through the question once more. We're saying that in this diagram that we have, it was an experiment that was used to determine the effect of a biological catalyst on the decomposition of what? Of hydrogen peroxide. Remember, hydrogen peroxide is going to decompose to form what? Oxygen and water. Now, the decomposition of this hydrogen peroxide, it can occur under several conditions, and one of the conditions is that there should be a biological what? Catalyst. Okay. Now, in this case, what was used as a source of that biological catalyst was the potato discs. All right. So the potato disc, or the potatoes, the source of that biological catalyst. Now, remember, in biology, we say that we have what are called enzymes, and enzymes we define them as what? Biological what? Catalysts. So the catalysts that speed up what? The rate of biological reactions, and these biological reactions, they can either be anabolic or catabolic, isn't it? Where we say that anabolic reactions are those building up reactions, and then catabolic reactions are breaking down reactions. So in this case, the kind of reaction that we are seeing here is a reaction that is involving the breaking down of what? Hydrogen peroxide to form what? Oxygen and water. Okay. So in other words, this kind of reaction that is occurring here is what? Is the catabolic reaction. Okay. Now, what are the conditions? What were those conditions that were involved? So there were three experiments that were conducted. So the first experiment that was conducted, there were no potato discs that were included in this that were added to the hydrogen peroxide solution. Then in the second reaction, or rather in the second experiment, there were 5 potato discs that were added, similar to the third experiment, where also 5 potato discs were added. Okay, and then the other condition was that whether these potato discs were boiled or they were not boiled. Now, the thing that we need to understand from there, from the concept that, remember saying that these potato discs, they are the source of our biological catalysts, isn't it? Which are the enzymes, right? So these potato discs, they are producing enzymes that are going to break down. So imagine if this is our, let me use a different color. Imagine if this is our hydrogen peroxide, and then that hydrogen peroxide is going to be broken down to form what? To form the water and the oxygen. So it's going to form those two particles, isn't it? Where we have the water and the oxygen. So now, this biological catalyst that we're talking about is the enzyme. Okay. Now, when we boil these potato discs, remember I said that enzymes, okay, the action of enzymes is determined, right, or rather is affected by things such as what? Number one, we have temperature. Number two, we have what? pH, isn't it? And also substrate concentration. Those are some of the things that and also there are also other things such as cofactors and so on, right? But in this case, what we are looking at, those effects, those things that affect those things that affect the action of enzymes, it's temperature. So you can see that in this case, we are applying temperature to the enzymes by boiling the potato discs. Okay, we are applying temperature to the potato discs. So when we apply this temperature, such that we boil the potato discs, what we expect to happen to the to these what? To these enzymes contained in the potato discs. What we expect to happen is that the enzymes are going to be denatured. Okay. So remember, we say that the higher the temperature, the higher the rate of what? The reaction. But there is what is called the optimum temperature for an enzyme. Once that optimum temperature is crossed, which is the highest temperature at which the enzyme is in at its peak, where it is in most active. But once that temperature is exceeded, what is going to happen is that these enzymes are going to be denatured. Okay, what it means when we say denatured, it means that number one, in terms of its form, okay, its structure, it's going to be deformed, right? It's no longer going to assume the same structure. Okay, so we see that in the first experiment we did not have any potato discs, right? So we can't say we boiled nothing. There was nothing to boil in the first experiment. In the second experiment, what we see, there were these 5 potato discs that were boiled. Let me use a black. So there were 5. Okay, so there were 5 of them, right? And then so in the second experiment they were boiled. Now what it means in this case is that here there were no enzymes. Okay, no enzymes, because there was no what? Potato disc. Okay, here again, because the potato disc has been boiled, it means that all the enzymes that were contained there, they have also been denatured, so it is as good as not having any enzymes. Then in the third one, that's where we did not boil. In this experiment, the potato discs were not what? Were not boiled. Now let's look at the result. What was the result? So you can see that the first experiment, how much oxygen was produced? Were the 1.5 cubic centimeters of oxygen produced. In the second experiment, what we see, also we had 1.5 cubic centimeters of oxygen. And what does this entail? Remember I said that in the first experiment, right? In the first experiment, we see that there was no enzyme because there were no potato discs. In the second experiment, quite well there were potato discs, but those potato discs were boiled, hence we had no enzymes at all. So this is why we are seeing that the first and the second experiment, they are producing the same volume of what? Of oxygen. Okay, they are producing the same volume of oxygen. Then the third experiment, where we are draw potatoes, we are seeing that we are having 10 cubic centimeters of what? Of oxygen. And this is why we say that enzymes are biological catalysts that speed up the rate of what? These biological reactions. So in this case, the enzymes contained in the raw potatoes, they speeded up the rate of what? The reaction, so that we had to produce this much oxygen compared to the other two reactions. Okay, so let's get back to the question that we have. So this is, remember, this is biology grade 12, GCE, right? GCE paper 2. Okay, paper 2. That's the question that we are answering, and this is question 1. Okay, so let's look at part A of the question. Part A, question 1. Part 1 says, what is the general name of a biological catalyst? We've already given the general name of a biological catalyst, right? We said that the general name of a biological catalyst is what? An enzyme. Okay, it's an enzyme. And then part 2 says, give a reason for boiling the potato discs in experiment 2. So the reason why the potato discs were boiled was to see the effect of temperature, or to simply denature, okay, to denature what? The enzymes in the potato discs. Okay, to denature the what? The enzymes in the potato discs. So that is part 2. Right, that is part 2. Let me probably erase part of the diagram so that I can write those answers. So part A, we're saying, but this is A, 1, we're saying that the general name is enzymes. Okay, then 2, we're saying that the reason why the potato discs were boiled is to denature the enzymes. Okay, to denature the enzymes. Number 3, it says, explain the difference in results obtained between the experiment. So we've already explained, we said that experiment 1, there were no potato discs. Okay, so experiment 1, there were no potato discs, hence no enzyme, right? Then experiment 2, the discs were boiled. Hello, are you there? The discs were boiled, right? Okay, so the discs were boiled, Hence, all enzymes were denatured. Okay, all enzymes were denatured. Okay, but experiment 2 had enzymes from the unboiled what discs. Okay, so that is the reason. So the reason is simply because experiment 1 and experiment 2 had no enzymes, but experimenting 3, so here's experiment 3, but experiment 3 had enzymes, right? That is the reason why we are seeing that difference. Okay, then let's look at part B. Predict what would be the result of one, increasing the number of potato discs in 5 cubic centimeters of hydrogen peroxide. Okay, so what would happen if we increase? Remember, we had what? We had we had 5 potato discs in 5 cubic centimeters of hydrogen peroxide. Now, what would happen is that in the case of the volume of oxygen that is being produced is going to what? Is going to increase in experiment 3. Okay? In experiment, so you can see that, remember, we said that the factors that affect the rate of the reaction is in one, we have these enzymes, the presence of an enzyme and also substrate concentration. So if we have the more enzymes we have and the more substrate we have, if we increase any of this, we see that what is going to happen, the amount of product, in this case oxygen being produced, is going to also increase. But what we are seeing in this case is that in experiment 1 and experiment 2, experiment 1, there's no potato discs that is being added, right? And then in experiment 2, all the potato discs, they are boiled, meaning that in that case there's not going to be any effect because the amount of hydrogen peroxide has remained constant, it is the same. So here in experiment 1, we still have 1.5 cubic centimeters. In experiment 2, we're going to have what? 1.5 cubic centimeters, unless if we also increase the amount of hydrogen peroxide. Let's say, for example, if we took it to, let's say 10, okay, let's say if it was 10 cubic centimeters, then we would expect that the volume, even here where there are no enzymes, the volume would increase by 2. So it would be maybe 3 cubic centimeters, and then it would be 3 cubic centimeters. But as long as the substrate, which is hydrogen peroxide, remains the same, it has remained constant, what is going to happen is that in the first and the second case, or other experiments, we're going to have the same amount of oxygen being produced. And then in the third experiment, where we have raw potatoes, all right, where we have raw potato discs, and then if we increase the number of potato discs, we are going to see that the volume of oxygen that is going to be produced in experiment 3 is going to increase. Okay, so we say that the volume, that is part B, okay, the volume of oxygen will increase in experiment 3, but constant in 1 and 2. So in experiment 1 and 2, it's going to be constant, it is going to remain the same. But in experiment 3, we're going to see that it's going to increase because we have increased the number of enzymes by increasing the number of potato discs. Is that all right? Okay. Let's look at part C, probably the last question of that. So suggest two other characteristics of biological catalysts not demonstrated in A and B of this question. So we've already described other characteristics, isn't it, of biological catalysts. So remember I said that biological catalysts, which are enzymes, number one, they are affected by what? By temperature, okay? they're affected by temperature. So it has been already demonstrated. We are seeing that in this case it was demonstrated. I mean, temperature by boiling those potato discs. Then the other thing that that has also been demonstrated is the concentration of what, of the enzymes. Okay, the concentration of the enzymes has also been demonstrated. So the other two that we mentioned is in the presence, or I mean the pH of the environment. Okay, the pH of the environment. So there are those catalysts, I mean there are those enzymes that act well, or rather that work well in acidic environments, and there are those that work well in what? In alkaline environments. So that has not been demonstrated in the previous questions that we've talked about. And then also the presence of what is called a cofactor. Okay, what is called a cofactor. In this case also, it has not been demonstrated in these questions that we've talked about so far. So the two characteristics of enzymes that we are going to mention at this point is, number one, the effect of, we say that enzymes are affected by pH. Number two, enzymes are affected by what? Cofactors. Okay, is that okay? Would we have answered our question? Okay, right. So that is about it, that question. Okay. So let's look at the second question of this paper. The second question of this same paper, and then we'll see what will follow. So it's very good that we are actually trying to prepare for your exam using past papers like this, because once we go like this question that we've gone through, this question is on enzymes. So at least from what we've discussed so far, we've discussed quite enough about it. The grade 12 topic on it, enzymes, isn't it? Okay. So of course, if we look at maybe one or two other questions on enzymes, we would say that we've probably covered the whole topic of enzymes. So let's look at this other question. So there's this diagram, that is question 2. There's this diagram. Okay. Right. So it's this one says the figure shows cells taken from a vascular tissue of a plant. Okay. So it says vascular tissue. So here we're talking about transport in what? In plant. Okay, that's what you're talking about, transporting what? Plants, right? So when we are talking about transporting plants, there are two important vessels that we talk about. Number one, we talk about what is called the phloem vessel, right? And then number two, we talk about what is called the xylem vessel. Okay, right? The xylem vessel. So those are the two important vessels that we talk about. Of course, when you talk about phloem, here we're talking about the vessel that transports what? Manufactured what? Food. Okay. So remember, after photosynthesis has occurred in the leaves, that manufactured food, which is glucose, okay, will be converted to a storage form, which is usually what is called starch, okay, and then it is stored, right? And that's why when you look at the leaves, there are times when you test a leaf for what? For starch. That is in the storage form. Starch is a storage form of glucose in plants. Okay, and then some of it is going to be converted to the transportation form. So there's a form in which glucose is transported. Glucose in itself is quite reactive and it is very soluble. So if it is being transported the way it is as glucose, we're going to find that it may not be able to reach where it's going. So it has to be converted to another form called what? Sucrose. Okay, so that sucrose is the one that is transported by what? By the phloem what? Vessels. Okay, by the phloem vessels from where it's being manufactured to all other parts of the plant. And then when you look at the xylem simply transports what? Water and absorbed mineral salts from the soil to all other parts of what? Or rather from the roots to all other parts of the plant. So once the roots, remember we have what are called root hair cells in the roots, okay, and then those root hair cells are going to absorb the water and the mineral salts by what is called active transport, isn't it? So of course, water is by osmosis, and then when you look at mineral salts, it's by what is called what? Active transport, being absorbed from the roots, and then it goes into the root hair cells, then it goes up, those substances go up the plant to all other parts of the plant. Okay, so that's basically about the phloem and the xylem vessels. Now, we have this diagram here, and the question is saying that this diagram shows a cell taken from a vascular tissue of a plant. Now, we need to identify, is it a phloem vessel or is it a xylem vessel, right? So we should be able to identify that. Okay. Now, when you look at the phloem vessel, remember I said that number one thing that we should know about the phloem vessel is that it has what is called a companion cell. All right, because these substances, some of them are being transported to all other parts of the plant by what is called active what transport. Now, the phloem cell itself, it does not have the capacity to produce or rather to carry out what is called photosynthesis. I mean, sorry, what is called respiration, okay, to produce energy. But it needs energy to transport those substances. Now, where is it going to get the energy to transport these substances? So it gets from what is called a companion cell. So a phloem cell has, rather a phloem vessel, the phloem vessel has more like stacks of these cells that are stacked together, one after another. And then between each of those cells, what we see is that there is what is called a sieve plate. A sieve plate has sort of holes or perforations, which are called sieve pores. Okay, so there are those perforations which are called sieve pores, so that it allows the movement of substances from one phloem cell to the next. So you can see that there are those holes that are left, or rather those spaces that are left for the passage of substances from one cell to the next one. So like from there, they will go into the next one just like that. Okay, so even there they were coming through those pores which are called the sieve pores. Okay, now this entire thing, this entire part is called the sieve plate. Okay, that is called the sieve plate. This is a phloem cell. I mean, this is the what? A companion cell, of course, within a nucleus there. Okay, and then this is it, the phloem cell, or what is called the sieve element. A phloem cell is also called a sieve element. Okay, then it also has a cell wall. Okay, because remember it is a plant cell, so because it's a plant cell, it should have what? A cell wall. Okay, so let's look at the questions that are there so that we go through it quickly. So identify the specialized cell. So the specialized cell in this case is what is called the phloem what? The phloem cell. Okay, the phloem cell. Okay, so that is A. B, I mean, this is A1. Hello, Gift, are you there? Oh, okay. You went so silent. I was thinking maybe you disconnected. Okay. Then part 2 says name one feature in the figure or rather in the diagram which enables you identify, okay, the specialized what? The specialized cell. Now there are several features here that should help us identify this what? This cell. So number one of those features is the presence of what? A companion cell, right? Presence of a companion cell. Okay. Number two, we can see that presence of the sieve, the sieve plate, and pores, right? And pores. Okay. So those are some of the features that probably helped us to identify this specialized cell. So if it doesn't have this companion cell and it doesn't have the sieve plate and pores, then we'll probably be dealing with something else. It's not a phloem cell, right? It is not. If it doesn't have those, we'll not call it a phloem cell. Then which letters on the diagram correspond to the following? Nucleus. So which letter corresponds to the nucleus? So of course we can see that it is what? A. So this is the nucleus of the companion cell, and then cytoplasm. Okay, which one corresponds to cytoplasm? Which one corresponds to cytoplasm? So the one which corresponds to cytoplasm is D. So that is the cytoplasm. So those same substances that are being transported, they are also the cytoplasm of that cell. All right. So D is the cytoplasm in that case. Okay. Now it would have been easy if this part, if B was reaching up to there. Let me use black. If B was reaching up to, let's say here, it would have been better to mention B as cytoplasm. But in this case, it's only going up to there, which is the cell wall. Okay. But in this case, the one which is the cytoplasm is D. Then part B of the question says, give two functions of the cell labeled B. Okay, give two functions of the cell labeled what? B. So what are the functions of the cell labeled B? What are the functions? So what did we call it? What did we call the cell B? We said that the cell B is what? The companion what? Cell. It's a companion cell. Okay. So what is it? The function number one. Number one, the function of that is production of energy. Okay, production of energy for the phloem, cell. Okay, for the phloem cell. Then number two is also for storage of cell organelles. Okay, so cell organelles are found within the what? Within the companion cell. So all those organelles like the nucleus. So you can see that the phloem cell itself doesn't have a nucleus, but the nucleus is found where? In the companion cell. So all those organelles that have different functions, okay? The different cell organelles with different functions, they are going to be found in the companion cell, right? And then part C, question C says, with reference to the specialized cell in the diagram, name the process by which substances are transported in the specialized cell. Now remember the fact that we are saying that it needs energy, okay? This cell needs energy. It means that the process by which these substances are are transported. So this was B, question B, and then we're talking about question C. So part A, part 1 rather. The function of this is, the process by which it is transported is what? Active what? Transport. I think I already mentioned that, active transport. Okay, so active transport is the process by which substances are transported in this specialized cell. Then, part 2 says suggest two substances transported by the specialized what? Cell. So what are those substances? Remember, we're talking about what? We're talking about basically food substances, isn't it? So we can talk about it. Part 2, we can talk about sucrose. Okay, we can talk about sucrose. We can also talk about things like amino acids. Okay, we can talk about amino acids. Okay, even lipids, all those substances are going to be transported by this phloem cell. Okay, so that was question 1 and question 2 of the grade 12 GCE paper 2 of 2016. All right, is there any problem, Gift? Okay, so can I give you questions that pertaining the what we've looked at today? Hello. Okay, so I'm going to end this video here.